I'm talking about the force of habit, new tests for morphic resonance. Morphic resonance is the idea that there's a kind of memory in nature. The so-called laws of nature are more like habits. Each species has a kind of collective memory to which each individual contributes and on which it draws. So there's memory everywhere in all self-organizing systems. The universe isn't governed by a kind of cosmic Napoleonic code that was there at the moment of the Big Bang, but by habits that have evolved throughout the evolutionary universe as a result of natural selection. Successful habits get repeated and become more habitual, unsuccessful ones die out. Well, this is a controversial hypothesis. I first put it forward in my book, A New Science of Life. This is the third edition of A New Science of Life, recently published, which in the US has been retitled Morphic Resonance. And the fullest exposition of these ideas is in my main theoretical book, The Presence of the Past, Morphic Resonance and the Habits of Nature. So I'm not here going to be explaining the whole hypothesis because I've done that in other talks. And if you want to know all the details, you can read about them in my books. What I am going to be talking about is experimental tests. This is a hypothesis, which means it's a guess about the way nature works. It's not a fact, because it's not yet an established fact that morphic resonance happens. In my opinion, it makes a lot of sense of the natural world. It explains a whole lot of phenomena better than the regular idea that the laws of nature are fixed and eternal. And it has many implications that enable us to interpret many phenomena in a different way. For example, morphic resonance plays a major part in inheritance. Genes are important. They enable organisms to make the right proteins. But that's what they do. They don't code for the shape of a face or the instincts of a spider or the ability of starlings to fly in murmurations in great flocks without bumping into each other. None of these things are made directly by proteins. The proteins are like bricks in a building or the cogs in a machine or something. They're part of something larger than themselves, and it's the way they're all organized that makes the difference. And that, I think, is what morphic resonance does through what I call morphic fields. In fact, it's known that genes account for only a small amount of heredity in most cases, and this is now called the missing heritability problem, and I think it's missing from the genes because a lot of it's happening by morphic resonance. It has many implications for understanding familiar phenomena like the similarity of twins. Morphic resonance depends on similarity. The more similar any two people are, the more they'll resonate with each other. And so identical twins, even if they're separated soon after birth, would continue to resonate with each other and show many similarities in their lives, not because these are genetically determined or programmed, as usually assumed, but because they're resonating with each other on the basis of similarity. I also suggest that our own memories work by morphic resonance. Our brains are similar to themselves in the past. We are more similar to ourselves in the past than we are to anyone else, which is why we resonate specifically with ourselves in the past and have our own memories. We're a bit like other people, so we also pick up collective memories from lots of other people. But morphic resonance, I think, helps to explain memory, which is usually assumed to be stored in physical traces inside the brain. Attempts to find those traces have failed over and over again, and I think the simple explanation is they're not there. Anyway, this is a very controversial hypothesis, needless to say. Some people opposed to it even denounce it as pseudoscience because it's not in science textbooks. any new hypothesis is not going to be in science textbooks until it's been tested more, and a consensus grows up that it really happens. And that's why a lot of my time and effort is spent in thinking about and trying to devise new tests for morphic resonance. There's already a lot of circumstantial evidence that supports it. For example, if you train rats to learn a new trick in Boston, Massachusetts, rats in Edinburgh, Scotland, and in Melbourne, Australia, should be able to learn the same trick quicker, just because the American rats have learned it. And actually, from a long series of experiments with rats in water mazes, exactly this phenomenon has actually been found. Large increases in the rate of learning that spread from Harvard to Scotland and Australia, and which didn't depend on the rats being descended from trained rats. All the rats of that breed seem to be getting better, and that's just what you'd expect with morphic resonance. The hypothesis also predicts that if you make a new chemical compound and you crystallize it for the first time, there won't be an existing form for that crystal, an existing lattice structure, because it's never happened before. But the more often you crystallize it, the easier it should get. And that indeed is what happens. New compounds get easier and easier to crystallize as time goes on, the more often they're crystallized. And it should be easier for people to learn things that other people have already learned. For example, computer programming or skateboarding or windsurfing should be getting easier to learn because so many people have already learned them. It's already known that standard IQ tests seem to have got easier to do over a period of decades without people getting any more intelligent. And this has been a big puzzle in the field of psychology. It's called the Flynn effect, after a psychologist called James Flynn who discovered it. And I think the reason is that not that people are getting smarter, they're not, but the tests are getting easier to do because so many people have already done them. The more that do them, the easier it should be for others to do them afterwards. There have been quite a lot of experiments on morphic resonance, and most of them have given very encouraging positive results, and those are summarized in the new edition of A New Science of Life. One example, for example, is an experiment done on TV, and this was done with puzzle pictures. This was done in the days before TV was online and you could see it anywhere. The puzzle pictures were shown in Britain and people were tested in Germany before and after. There were two puzzle pictures, only one was shown on TV. The question was, did that one become easier for Germans to see after British people had seen it, compared with the control picture? And what I mean by puzzle pictures is, I can show you here, this is one of them. As you see, this picture is made up of black patches, and most people can't see the underlying image within it. But maybe you will if a small percentage can, but it's very hard to spot. Now, if I show you the answer, you see it's a cowboy on horseback. And when you look at the puzzle picture again, you'll see a cowboy on horseback. And you'll just get it straight away. And if you're shown this picture in 5 years' time, you'll probably get it straight away. It's a very, very rapid form of learning. Exactly the same light is going into the eye, but the way you interpret it is different because of your experience in seeing the answer. Now, in this experiment, when people in Britain, several million people in Britain, had been given the correct answer on TV, they were shown the puzzle picture, it melted into the answer and melted back to the puzzle picture, and they got it. After that, people in Germany found it easier to recognize compared with the control picture that hadn't been shown on TV here. So that's one example of a morphic resonance test, and there have been quite a number of others. But what I'm interested in doing is finding out new ways of testing morphic resonance that are easier to do. It's hard to organize experiments involving television, and today, with everything being streaming online, it's really hard to do that type of test anymore. One area I'm particularly interested in is in the realm of new materials. Just as when you make a new chemical, the more often you crystallize it, the easier it should get to crystallize. If physicists and chemists make completely new kinds of material, then if they're materials that show a coherent structure, an integrated pattern of activity, the more often they make them, the easier that pattern should be to make appear. It should appear quicker or easier or stronger the more often the experiment is repeated. And at the moment, there's a huge amount of creativity in materials science and in solid-state physics. I subscribe to the international scientific journal Nature, and some of the most interesting papers each week are those in some of what I think are the most creative areas of science at the moment, solid-state physics and materials science. And I collect them. I have here a sheaf of papers from recent issues of Nature dealing with these new materials. And it's just astonishing the kinds of things people are finding out. So here, for example, is a report called The Sound of a Hidden Order. Ultrasound measurements in copper oxide superconductor have revealed an exotic phase of matter composed of loops of spontaneous quantum currents that has hitherto excelled in evading observation. Now, I think that these spontaneous quantum currents would, and the overall patterns of this exotic phase of matter would emerge more readily, more quickly, and the more often this is done. So people working in these areas could look to see whether there's any change in the behavior the more often they make these new forms of matter. Here's another one, a reprogrammable mechanical metamaterial with stable memory. And again, a completely new kind of substance, a metamaterial. Here's another one. Evidence for unconventional superconductivity in twisted bilayer graphene. Superconductivity is when the electrons get into a coherent state that spreads over a whole area of material, and they can conduct electricity without friction. This is very different from electricity going down a normal wire. It's a coherent quantum state. And again, I think that it should be easier to make these the more often they're made. Here's another one. Antiferromagnetic half-skyrmions and bimerons at room temperature. All of these, and many, many more, and these are just recent ones, are new materials, new states of matter that are being explored in laboratories. And they should show morphic resonance effects. The people doing this research don't usually look for morphic resonance effects because that's not what they're looking for. They're characterizing these materials. And if they find that the properties appear more clearly or the substance appears more easily as they do the experiments over and over again, they almost always assume that it's just them getting better at doing it. They don't do the same thing under exactly the same conditions over and over again to see if the material itself is changing, if the phenomenon itself is changing. So even if they notice changes, they usually assume it's them getting better at doing it. So what I'm suggesting is that they pay attention and really look out for morphic resonance effects. And if they do appear in any of these materials, they could even have technological applications, particularly in quantum computers. There could be a kind of built-in memory in these systems with global link-ups with other similar quantum computers by morphic resonance. An entirely new technology could emerge from this. Well, taking another possible kind of test, thinking of word puzzles. Now, with crossword puzzles, years ago, various people wrote to me and told me they'd found with the Times crossword puzzle, they found it easier to do if they did it in the evening or the next day compared with first thing in the morning when the Times arrived, their hard copy newspaper. Many other people have reported this with other newspaper crosswords. And the possibility there, you see, is that the crosswords get easier to solve during the day because so many people have already solved them. And so therefore they might indeed be getting easier. And years ago, a student at Nottingham University in England did an experiment with crosswords. In those days, newspapers weren't online either. She used the London Evening Standard newspaper crossword. The Evening Standard kindly provided crosswords in advance of publication. And she compared people's ability to do that crossword compared with one that had already been published as a control. She had different groups of people before and after the crossword was published in London. And sure enough, the ability of people to do the test crossword increased relative to the control after lots of people had done it in London. Well, I think there's a wonderful opportunity for doing research of this kind through new word puzzles which are available online. I'm thinking particularly of Wordle, which has become an internet sensation. It's a five-letter word, and you have to guess which letters make up the word, and you have 5 goes, and if you haven't done it, you've failed the test. This is quite addictive, and some people love doing this puzzle. My own wife is very keen on Wordle. Now, on the morphic resonance principle, Wordle is published every day. There's a new one published at midnight, and those who do it first might find it harder than those who do it towards the end of the day, because so many people have solved that puzzle already. So Wordle could give a test for morphic resonance day after day by looking at the statistics of people's success rates as time goes on. Now, if there is an increase in success rates as time goes on, then obviously the skeptics, and indeed I myself, would say, well, could this be sensory leakage? Could it be people sharing the answer with others, or hearing other people talking about it, or picking up clues from people who've already solved it? That indeed is possible, and skeptics who don't believe in morphic resonance would probably predict that these scores would go up throughout the day because of some kind of leakage of information. Well, obviously for a morphic resonance test, it would be important to rule that out. So you'd have to have subjects who were not exposed to other people's answers on Wordle, and perhaps who were not connected up online all day and are tested at different parts of the day where they're not in contact with others, so they can't have picked up any of the clues by sensory leakage. These are technical details, but it should be possible to do this research. And indeed, I've tried. My son Merlin has a friend who works at the New York Times in a senior position, and so through him I managed to get in touch with people in the New York Times, which now owns Wordle, and got through to their games division to see if they would be prepared to cooperate in this research. And unfortunately, they weren't. I think it may be partly because they don't have the data accessible in a suitable format. Anyway, unfortunately, they weren't cooperative. But there are various other puzzles like Wordle. There's one called Quordle, and then there's one where you do 4 words, and there's another with 8 words, another with 16 words. These are not owned by the New York Times, they're run by other people. And if you happen to work for one of these companies or know someone who does, the first thing is to find out whether they retain the data throughout the day from different people's success rates. And secondly, to look at the data and show whether they do show a rising trend of success throughout the day. If so, this would make a wonderful and fun way to do research on morphic resonance with a new experiment possible every single day. And one could then look at various variables and things that might influence the process. Well, that's one test in the human realm, and one can think of many others, and indeed do think of others if you can, and especially if they're ones you could put into practice. These experiments don't have to be particularly expensive. It's best to ride on the coattails, as it were, of things that are already happening, like crossword puzzles or Wordle, because if you had to recruit millions of people to take part in these tests, it would be a hard job recruiting them. But if you find something that people are doing anyway, then it makes it much easier. I now want to turn to another kind of learning that could be tested by morphic resonance, namely conditioned aversion. Attraction and aversion to foods are things that are learned very fast indeed, and this is not just in humans, it's in many species of animals. If you eat a new kind of food that you've not had before, and you're sick soon afterwards, you probably won't ever want to eat that food again. It'll really put you off. This happened to me once when I was at school. I somehow got hold of a large amount of fudge, which is made out of brown sugar, and I ate lots of this fudge to the point where I was actually sick. And as a result, I've not been able to eat fudge ever since. It created this conditioned aversion in me to fudge. So this is a very, very rapid form of learning. And somewhat less rapid, but also very fundamental in animals, is attraction to particular foods. And this is something animals can learn, that a particular smell is associated with food, as in Pavlovian conditioning. As Pavlov showed, dogs can associate the sound of a bell with being fed, and they start salivating when they hear the bell ring, when they've got this conditioned reflex. And the same happens with flavors or tastes with food. And you can see these are very fundamental to animals, and because they need to avoid harmful foods and they need to be moved towards nutritious foods. Well, in the late 1980s, I did an experiment on conditioned aversion in day-old chicks. This happened in the laboratory at the Open University of Stephen Rose. And in 1988, Stephen Rose and I both wrote for the Guardian newspaper. I had a monthly column, and he also wrote there quite often. And I wrote a column about memory not being stored in the brain. Well, Rose is a materialist and believes memory is stored in the brain, and was very opposed to my idea that it couldn't be or wasn't stored in the brain. And we had a controversy in The Guardian, went on for several weeks, backwards and forwards. And at the end of it, he challenged me to test what he called this seemingly absurd hypothesis in his own laboratory, under his own supervision. Well, various friends of mine said that I'd be stupid to take up this challenge because he could control all the details and I'd be entirely at his mercy. But on the other hand, no one else had offered to do an experiment in their laboratory because morphic resonance is so controversial, most people want to steer clear of it. So I accepted Rose's challenge, and he at the time was working with day-old chicks, and he was training them to be averse to particular stimuli. What he was doing was getting them to peck at little LED bulbs. So if you had a little blue bulb, when a day-old chick will peck at any shiny object, because it might be food, and the chicks would peck at the blue LED bulb, and then if nothing happened, they'd peck at it again, and so on. But if after pecking at it once, you gave them an injection of lithium chloride that made them sick, then they wouldn't peck at it anymore. They'd develop an aversion to that stimulus. Well, in this experiment we designed together, we had a control stimulus, a chrome-plated bead, which was just shiny, and a new stimulus, a yellow LED, light-emitting diode, little bulb, which chicks hadn't been trained on before. After the control stimulus, they were given an injection of saline solution, the control, which had no effect on them, beyond giving the injection itself. And after the yellow LED, they had an injection of lithium chloride that made them sick, and they'd never touch yellow LEDs again. What we did was the same experiment. It was done by a student. We did the same experiment day after day with fresh batches of day-old chicks. My prediction was that relative to the chrome bead, they'd become more and more reluctant to peck at the yellow LED because previous generations of chicks had found it made them sick. And what we did was measure the time it took them to peck at the LED, and it's called the latency, how long the delay was before they pecked it. Some didn't peck it at all. And I predicted that relative to the control, the latency would increase. Rose predicted that it would not. And we did the experiment, and the results you can see here. This shows the time along the bottom and the latency along this axis. As you can see, as time went on, the latency, the time taken before they peck, average time taken before they peck at the yellow light-emitting diode, went up. And this was a highly significant effect statistically. Well, Rose didn't know what to make of this, and the only thing he was sure of was that it couldn't be morphic resonance. It must be some kind of artifact, because he thought morphic resonance is impossible. And he first of all got me to agree that we'd publish the results together, whatever happened. And when, unexpectedly, it gave a positive result for morphic resonance, he then didn't want to publish them together, so I published them. And he then wrote a response saying why he thought they could be explained in some other way, and I wrote a response to him. So this illustrates the fact that in science it's not just a question of evidence, it's how you interpret the evidence, and people who don't like evidence will always try and explain it or explain it away in some other way. So I can't claim this is conclusive evidence for morphic resonance because Rose has put forward alternative arguments which I've refuted, but nevertheless this is not going to be accepted as clear evidence on just one experiment. But for me it's very encouraging, and I think what's needed is a new series of experiments on conditioned attraction or aversion. One suitable way of doing this would be with fruit flies, Drosophila, which are very commonly used in laboratories. They're extremely cheap. You can breed the flies for almost nothing. And there's already a form of test that people do for conditioned aversion in fruit flies or conditioned attraction. You have a T-shaped tube, a glass tube, with two arms going that way. You have air gently blown in from both arms, and the flies move up the tube, and when they get to the T-junction, they can go that way or they can go that way. And if there's an attractive chemical here, then most of them will go that way. If there's a repellent one here, most of them will go that way. So this is, already apparatus exists for this. You can quantify the response. And so it should be relatively easy to do experiments where Drosophila, fruit flies, are made averse to a particular smell, or they're attracted to a particular smell because it leads them to food. And then, as in the chick experiment, the same experiment is done over and over and over again with fresh batches of fruit flies whose parents have never been exposed to this apparatus. It could also be done with ones whose parents have been exposed to it and may well show an increase in aversion or attraction, which could be epigenetic, an inheritance of acquired characters. And epigenetic inheritance is now widely recognized in biology. It means that organisms can inherit what their parents have learned or adaptations their parents have made. So it could be partly due to morphic resonance, it could be partly due to molecular changes in the expression of the genes. But in this setup I'm talking about, it's possible to compare the effects of, say, 10 generations of training, whether it makes them more attracted or repelled by a chemical when their parents have been trained compared with when they haven't been trained. If the effect shows when the parents haven't been trained, they're just naive animals, then that would be a morphic resonance effect. If it shows up when they've been trained and where the parents have been trained, it could be molecular epigenetics combined with morphic resonance. But this experiment of comparing pure morphic resonance with epigenetic inheritance would give some measure of what the contribution of morphic resonance is to the epigenetic effect. So this is a very simple experiment with fruit flies, and similar experiments could be done with a tiny nematode worm called Caenorhabditis elegans that's used in many biology laboratories. Even easier to do really than with the fruit flies, because these worms wriggle in petri dishes. You don't even need tubes and airflows and things to do the experiment. So similar experiments with conditioned aversion or attraction can be done with worms. It's already known that many insects, in their preference for food plants, are influenced by what previous generations have experienced, either toxic or non-toxic effects. It's already known that conditioned aversion has mysterious effects on rats. In the rat poison industry, it's well known that if you poison rats with regular poisons, very soon they become what's called bait shy. You can kill some of the rats, but soon other rats will just stop avoiding this poisoned food. Even in different places, this spreads, this bait shyness, in a way that I think is because of morphic resonance. It's a really big problem in the rat poison industry, which is why the most commonly used rat poison now is warfarin, or similar substances that prevent blood clotting. And the reason they work is that you can poison the rats with warfarin, and it takes several days before the effect comes on, so they don't learn to associate it with a particular food or bait. And it's precisely because of that that warfarin's used, because bait shyness is such a problem, and as I say, I think that involves morphic resonance. Harder to do the experiments with rats and rat poison than with Caenorhabditis nematode worms or with fruit flies, which is why I suggest doing it with fruit flies or worms, where you can get through many generations in a couple of months, whereas the rat experiment would take much longer. And also, of course, most people wouldn't particularly want to be working with rats that they're poisoning. Now I move to another way of testing morphic resonance, in this case in cell biology. And this is based on a phenomenon called the Hill effect, discovered by a French biologist called Miroslav Hill, working at one of the French state biology laboratories. Hill discovered that if you take normal tissue culture cells, mammalian cells in tissue culture, and you grow them in a toxic medium, or you grow them in extremely high temperature, that's the limit of what they can stand, after a number of generations, some of these cells acquire the ability to survive in these conditions. Now, he discovered this through experiments in which he subcultured the cells from petri dishes or test tubes from the normal medium, from normal medium to normal medium to normal medium, generation after generation. But some of them he put in a medium containing toxins, or at high temperature, the shaded circles you see there represent the stressed conditions. What he found was, after a number of generations, they started surviving in these stressed conditions. And once they'd started surviving, generation after generation, they continued surviving. They got more and more resistant to the toxin or the high temperature. And he thought this must be because of mutations, but there was no way in which the stressed cells could influence the line that's being propagated because they weren't being bred from. The only cells being propagated were ones that had never seen the stress, and yet they were responding as if they had, as if they'd benefited from an adaptation acquired by the cells in the stressed petri dishes. Well, Hill tried to explain this. He was very baffled by it. He repeated the experiment with different toxins, different kinds of cells, and different heat stresses over and over again, and found it was a repeatable phenomenon. He didn't call it the Hill effect. Subsequent people call it that because he was the person who discovered it. And his interpretation was that in some way the cells are somehow entangled through some kind of quantum entanglement. So the ones that have learned to adapt to the stress modify the ones that have never been exposed to it, which pass on the modification to the next generation, which enables them to respond to the stress better. Well, there's no evidence for quantum entanglement in biological systems of this kind, and he admitted this was a very speculative explanation. But my own explanation, in terms of morphic resonance, is different. I think that the cells that have learned to adapt could directly influence subsequent batches of cells that are adapting to the same stress by morphic resonance, and the more that have done it, the greater this influence would become, the stronger the Hill effect would appear to be as time goes on. Well, I think this is a very fascinating experimental system because, apart from anything else, it tells us about the biological ability to adapt to stresses. And this is of great importance in ecology, climate change, response to toxic chemicals in the environment, or even the deliberate breeding of organisms that can deal with toxic chemicals in waste dumps and in the environment. So I've proposed an experiment to test the Hill effect, which is summarized in this diagram here. In one case, the cells are treated as Hill did in every generation exposed to the stress you see along the lower line. But in the upper line, for the first, say, 4 or 5 generations, they're not exposed to the stress, and then they're exposed to the stress. Now, his entanglement theory would mean that the ones on the top line wouldn't be entangled by stress cells for quite a number of generations, however many you choose to do it without the stress. And then when you start exposing them to the stress, it might take 5 or 10 generations, if that's what it took the first time round, for this effect to show up. But if morphic resonance is at work, it should show up much quicker. You shouldn't have to do it so often. It should happen more readily, even though the cells you're testing are not entangled, quantum entanglement, with those that have already been tested. Now, this may sound a little bit complicated and technical, but actually the experiment in principle would be quite simple to do. And it has in fact been replicated once in an experiment in Hyderabad, India, which I helped arrange using mammalian aortic cells in tissue culture and a toxin called thioguanine. And after 27 generations, the cells did start surviving and flourishing despite the stress. It took a long time because we used an excessively high dose in that experiment. But these experiments could be done with both mammalian cultured cells, as Hill did. They could be done with yeast cells. They could be done with bacterial cells. And they could be done in quite simple laboratory conditions. You don't need a very, very advanced lab to do this. In fact, even in a school lab, you could probably do these kinds of experiments. So this is a test I'd very much like to see done, and in fact, all of these I'd like to see done. And I think these could provide simple, straightforward, relatively straightforward ways of providing new tests for morphic resonance. First, let me summarize. First, by looking in labs in solid-state physics and in new materials science labs, where people are looking at new materials to see whether the properties of these materials, when the experiment's repeated under similar conditions over and over again, whether the properties appear more strongly, more quickly, more easily, as would be expected with morphic resonance. Secondly, experiments with word puzzles or other puzzles which are online and in which many people participate to see whether scores get better throughout the day under conditions in which sensory leakage or people sharing the answers can be ruled out. Thirdly, tests involving conditioned aversion or attraction in invertebrate organisms like nematode worms or fruit flies, where the experiments can be done quickly. And fourth, an attempt to replicate the Hill effect using cultured mammalian cells in laboratories, or yeast or bacteria or other cells, showing that adaptations can pass from one cell to another by morphic resonance without the need for genetic or any other kind of material continuity. All of these, I think, could help open up the field of morphic resonance research, which would in turn lead to a revolutionary change in the way we understand biology, chemistry, physics, and indeed all nature.